Hi everyone, this is Ellen George with Great Lakes Cisco, and I'm at the 13th International Coregonid Symposium in Bayfield, Wisconsin. And we're going to be talking with Hannah Lachance from University of Vermont on some of her work with the effect of light on Cisco gene expression. So over to you, and go ahead.
Hi. As Ellen said, my name is Hannah, and I am from the University of Vermont. And we're doing a project that looks at how climate change, more so the effect of climate change on winter lake conditions, will affect Cisco egg development. So first of all, there's kind of two main environmental components that we're focused on, temperature, but also light. And not a lot of research has been done on light before, so we started with a pilot study to look at how light will impact egg development. And so we started with some kind of extreme treatments just to see what's happening or if anything's happening. So we raised Cisco under 24-hour light, 24-hour dark, or a regular photo period as our three treatments. And some of the results are presented earlier. Go check out Taylor Stewart's presentation, where he shows that there was a week difference between the 24-hour light and the other two treatments. They hatched a week earlier, the 24-hour light. And the yolk sac size differences as well, which is more Taylor's side of the project. I'm looking more so at what's happening on the molecular level. So what are those differences that we're seeing? What does that actually mean? And unfortunately, I don't have any results to show you right now, but stay tuned. I'll have some soon. But based on the literature, we kind of expect to see differences in circadian rhythm genes, more specifically the clock genes, and also stress response and DNA repair genes have been shown to be affected by light. So now that we know that there's probably some changes at the effect of light on egg development, we're moving forward with our full factorial design where we incorporate 3 levels of temperature and 3 levels of light. So that will be with eggs from Lake Superior right out the window here. And we will be raising them under the different treatments. We'll have 6 replications for each treatment. And we will also do another genetic analysis. What that entails kind of depends on the results from the first project. And we hope to find something interesting and kind of figure out how this can impact their future in the lake, but also just help us understand what's going on with the changing environment. So
great.
That's kind of my spiel.
Yeah, great. I liked Taylor Stewart's presentation. If you are interested in that, you can watch the live stream on that, also archived on Periscope. And I know, and one of the things that he found is that you mentioned is that the eggs raised under 24-hour light hatched a week earlier, which in a species like Cisco, where the timing is so tied to ice out and it's such a quickly changing environment, like a week doesn't seem that long, but it really can be long in the scheme of Cisco, life.
Yeah.
But like the difference between a normal photo period and 24-hour light is pretty extreme,
right?
And so there was like an 8-day difference, I guess, is what we could consider maybe the most extreme change.
Right.
So if this is kind of tying yours and Taylor's together. So if Taylor finds that maybe
I just want to make a quick announcement.
We're getting a quick announcement.
If you are planning on not going on the cruise, if you have a ticket and you're not going on the cruise, please give your ticket to a person in a blue shirt. And they have tickets available for anyone who would like to go on the cruise who does not have a ticket. No, they don't. I see a head shaking.
No one wants to give up their cruise ticket.
It's a pretty sweet gig, I guess.
Yeah. Okay, thank you for that little commercial break. Anyway, so what I was saying is that trying to get at, I guess, with my question is a couple day difference. Do you think that would be ecologically relevant? Like, say the amount of light that they're actually going to experience in the wild Do you think if that changes in a couple days, would that be ecologically relevant? And how would your work on genetics maybe inform our decision on whether or not it's relevant or not?
Right. So there's a couple different answers to that. One that Taylor talks about a little bit more is kind of the match-mismatch hypothesis that focuses more on prey availability for the larvae when they hatch. Some impacts that I could be seeing at the genetic level, like I said, we expect maybe differences in development because of throwing off the circadian rhythm. But there's also been evidence that it can affect stress response and DNA repair genes, and that kind of ties into longevity. Like, they might not die right away, but they might not be well equipped to survive well into adulthood. And so perhaps that's where, you know, that kind of critical first year could be impacted by these kind of advancing the development past what it requires to fully express all the genes and fully, you know, kind of turn those genes into proteins and work through the mechanisms that it needs to. So again, at this point, this is just based on the literature. It'll be really interesting to see what we actually find, and that should inform kind of moving forward the light intensities that will be more realistic examples of what the varying levels of ice and snow cover and lack of ice and snow cover will actually entail for the Cisco. So
I think it's really interesting that, like, in humans, circadian rhythm and effects of light has been studied a lot on its effects on prolonged stress and kind of long-term stress. So it'll be interesting to see what you find as well, and kind of bring the idea of early hatching and hatching time out of just like zooplankton match-mismatch and into something else.
Exactly, because another area is, you know, Taylor talked about the yolk sac being perhaps overutilized during egg development because of the accelerated development. So that's, you know, kind of a little bit concerning. They may not have enough to live off of. And I don't know if he presented on this, but we did actually see a decline in the larval populations after hatching in the 24-hour light. Most of them died off.
Oh, so, really? Okay.
Yeah. I don't know if that's like public knowledge, but it is now.
Yeah, it is now. It's a preliminary finding.
Right, preliminary. But
well, that's awesome. I'm really excited to see the results from this project. So,
thank you.
Thank you, Hannah. And if anyone is watching this later, if you have any questions, you can comment on the video and I can relay them to Hannah, or you can tweet at her. She's at Hannah M. Lachance. She's one of our Cisco tweeps, I guess. And I'm really excited that she has her handle on the poster. So good job there, Hannah.
Thank you.
Thank you very much. And I'll see you later.
Thanks.
Bye.
Bye.